Hi everyone, my name is Tensui. Today I'll be presenting my protein model of Piezo1. Piezo1 is a magnosensitive ion channel located in a plasma membrane of many cells. It is especially important in red blood cells and endothelial cells, which line blood vessel. So Piezo1 allows cells to detect magnetic force, such as membrane stretch and blood flow. Mutation in the Piezo1 protein can cause a disease called dehydrated hereditary stomatocytosis, which affects the hydration and shape of red blood cells. Here is the overall 3D shape of the protein. This model represents the three-dimensional structure of Piezo1. Piezo1 forms a trimer, meaning it's made of three identical subunits that assemble together to form a functional ion channel. In my model, the three arms made from straws represent the three subunits of the protein. The center of the model represents the ion pore, which is the opening through which ions such as calcium ions can enter the cells. And now let's talk about the protein structure levels. Different part of my models represent different levels of the protein structure. First, the primary structure is represented by these beads. Each beads represent an amino acids in the sequence of the protein. And if I spiral it, as you can see this one, and the blue one, the green one, when the beads are arranged in the spiral shape, they represent an alpha helix, which is the type of secondary structure formed by hydrogen bonding within the polypeptide chain. And if I fold the entire straw, the overall three-dimensional folding of a single straw represents the tertiary structure, which is the 3D structure of a single protein subunit. Finally, when the three subunits assemble together, this forms the quaternary structure of the Piezo1 protein. Now let's talk about mutation. This beads here, I used the red one. It represents the mutation site in my protein. The mutation is called R2456H, which means that the amino acid R at the position 2456 is replaced by histidine. This mutation occurs near the region that controls channel inactivation, which is the process that normally closes the channel after it opens. The normal function of Piezo1 is a magnosensitive ion channel. When the cell's membrane experiences magnetic force, such as stretching or pressure from blood flow, like these three straws will sense the pressure and the channel will briefly open. I can pull it out a little bit. This allows positive charge ions, especially calcium ions, to enter the cell. After a short time, the channel closes again, which helps maintain normal ion balance inside the cell. In red blood cells, this process helps regulate cells volumes and hydration. And the mutation, the R2456H mutation, was the mutation the channel does not close properly. It will slow down the whole process of channel inactivation, means that the channel will stay open longer than normal. As a result, too much calcium will enter the cells, and in the red blood cells, this will trigger potassium loss and water loss, which causes the cells to become dehydrated and abnormal in shape. And this will lead to disease we mentioned at the beginning of the video. Because the mutation caused the channel to remain more active than normal, this mutation is classified as a gain of function mutation. There's some limitation of my model. First, the model is not to scale because the real Piezo1 protein contains over 2,500 amino acids per subunit, and it's much more complex. Also, it doesn't show the atomic interaction such as hydrogen bonding or other interaction between amino acids. And third is that it's static. Real protein are dynamic and they change shape during activation and inactivation. In summary, Piezo1 is a magnosensitive ion channel located in the plasma membrane that allows cells to detect magnetic force. Mutations such as R2456H disrupt the normal regulation of the channel, leading to excessive ion flow and disease. Understanding these mutations helps scientists better understanding how magnetophores regulate cellular function and how defects in these systems can how defeats in this system can lead to human disease. Thank you.